Hi, and welcome to this lecture on environmental adaptation of animals. This is the last video on molecules and cells in animal physiology and dealing with receptors. Something that you already know, at least something, but it's so important thing in animal physiology that it's nice to have a review. So, the metabolic pathways are regulated by different receptors, and these receptors are located in the cell membrane to detect that, okay, are there any ligands available there in the extracellular fluid, or then they can be in the nucleus. And we have several routes that can be used by these receptors. We have receptors that are working as an ion channel. In that case, the response is on the electricity. Then we have G protein mediated receptors, then the response on G protein, surprise, surprise. And then the receptors can be autophosphorylating. So they are phosphorylating themselves or phosphorylating something else, etc. And then we have different kind of signaling cascade that is causing the end product. And it can contain phosphorylation, or then it can have some secondary messengers. Cyclic AMP, cyclic GMP, inositol triphosphate, diacylglycerol. And in case of ion channels, well, they are ligand-sensitive ion channels. So when the ligand is touching the receptor, there is a pore that opens, and then depending on the selectivity of the ion channel, they either permit only one type of ion, or in this case, they can permit practically everything. So in the typical mode, these ligand-gated channels open to permit the ions when they bind these ligands. And then when the ligand is moving away, then this ion channel is closed. And then it's affecting on the electric charge across the membrane, meaning electricity. Then the second way is to have these G proteins. We have a G protein coupled receptor where the ligand is binding, and then making this receptor activated. And when it's activated, it's interacting with two different cell membrane proteins, a G protein and then an enzyme that is activating these intracellular enzyme catalytics. So when this G protein mediated receptor is activated, it's activating G protein. And then this G protein is moving on the surface of cell membrane to this enzyme, and then this enzyme is producing from ATP to cyclic AMP. So it's used as a secondary messenger. And then this AMP, this secondary messenger, can be distributed inside the cell to activate or modulate the activity of certain different processes. Then we have cyclic GMP that is modulated directly with this receptor. So ligand is binding and it's activating the catalytic side of the same molecule and then producing cyclic AMP from GTP. But because now there is only a little bit limited, it's limiting also the number of cyclic GMP produced, because now it's done in the same molecule. And then we have these transcription factors where the ligand is moved inside the cell because it's hydrophobic. And it's also moved into the nucleus and binding on the intracellular receptor on the DNA and affecting on the transcription, and then affecting that, okay, some genes are upregulated or downregulated. So we can have different kind of ways. We have these G proteins that are affecting to produce a cyclic AMP, and then we have different kinases that are regulated, and then we have phosphorylation of different proteins. Then we have this GTP that is producing cyclic GMP and pretty much the same kind of responses. Then we have nitric oxidate that is distributed directly through the cell membrane, for example, to produce cyclic GMP. And some G protein coupled receptors are operating via phospholipase C to produce diacylglycerol that is affecting on different kinases, but also it's releasing IP3, so inositol triphosphate, that is affecting on calcium release from intracellular sources, and then this calcium is activating different kinases. So we can see that, okay, we have several different ways to regulate them, and quite often in the animal physiology we will deal a lot of different of these processes. Okay, what about then the example? For example, the glycogen cleavage that we also already had in the previous video. So when the receptor is activated, we have this G protein activation. And actually, we can have several G proteins activated. And then we have activated adenylyl cyclase to produce a lot of cyclic AMP. Then when we have inactivated cyclic AMP dependent kinase, it's now activated because now we have this cyclic AMP. And then we have this kinase activated. It's activating this glycogen phosphorylase, and therefore we have now this active glycogen phosphorylase kinase that is then used to activate the glycogen phosphorylase. And that's why, even with a single molecule of epinephrine, meaning adrenaline, so we have a huge number of active glycogen phosphorylase. They're cutting glycogen to glucose. And so the amplification happens in several steps, and that makes it very efficient. Thank you.